So this is Rowan, Cosmo, and Helen from San Francisco Friends School, and we're at Table A.
Our science experiment was about salt. Our question was, how do different amounts of different types of salt affect bacteria? This question is important because it will show how different types of salt can be used to prevent bacteria growth. Also, salt is used often, so it's good to know which salt is better. Our hypothesis is that if we take different types of salt and put them on bacteria, then they will all kill the bacteria, but slightly differently for each one, because all salt kills bacteria, but the different kinds of salt are processed differently, so they will probably kill the bacteria slightly differently. This is important because we can find out which salt is the better bacteria killer. We thought that if there's like 1 pinch of salt or not that much salt, then most of the bacteria will die. But if there was more salt, then almost all of the bacteria will die, and the only bacteria remaining will be some bacteria that responds well to high salinity, which you can see in the research paper below.
This is our expected graph, and it shows that with 0 pinches of salt, obviously no bacteria will be killed. With 1, we think that 75% of the bacteria will be killed. 2 pinches, we think about 95% of the bacteria will be killed. And with 3 pinches, we think all of the bacteria will die. We did an average instead of each salt because we think the salts will be very similar, enough so that we didn't need to show each. This is our research paper, and in our research, we have found things about salt working against bacteria. For example, kosher salt can help remove the bacteria that causes salmonella. The website that says this is right there. We also found a website about bacteria that responds well to high salinity levels, which is right here, and that bacteria is called halophilic bacteria. This is our bibliography. It shows the link, the author and the publisher, what was in there, and some details about the article we found. 4 good sites.
This is our materials chart. It tells us what we need, why we need it, where we're going to get it, and sort of details about it. And we used sea salt, kosher salt, and iodized salt, and other things we needed were petri dishes, Sharpies, cotton swabs, and an incubator. And this is our procedure. The first bit basically tells you how to label, streak, and seal the petri dish with parafilm. And the second part tells you how to put salt on the petri dish after you let bacteria grow for a little bit. And an important part is to not put the petri dish when it has salt on it back in the incubator, because that will let more bacteria grow, and that's not what we want to do.
Okay, this is our data table. It shows that with 0 pinches of salt, no bacteria was killed at all. As you can see from the averages section, it followed the trend from our expected graph. It continued up at an almost steady pace, killing the most in the 3 pinches and the least in the 0 pinches. Sadly, the salt did not kill nearly as much as we thought it would.
And here is our final graph. The blue line is sea salt, the green line is the average, the red line is kosher salt, and the yellowy orange line is iodized salt. It is interesting because the average is very close to the kosher salt. One trend that we saw was that iodized salt is always killing the most amount of bacteria, kosher salt in the middle, and sea salt isn't very good, I guess. Maybe because it doesn't have anything added, like iodine. So that was our graph that we saw, and that is our experiment.
Thanks. Goodbye.